Hello, everyone. Today we'll discuss Western blotting, a crucial technique in cell and molecular biology for protein separation and detection. It allows us to isolate and identify specific proteins from complex mixtures, like cell lysates. The procedure involves separating proteins by size, transferring them to a solid support, and detecting the target protein using antibodies. SDS page is often used for macromolecule separation, with sample preparation being its initial step. Therefore, protein samples are treated with a loading buffer containing SDS, beta-mercaptoethanol, bromophenol blue, and glycerol to provide a uniform negative charge to all proteins. After adding the loading buffer, the samples are heated to 95 degrees Celsius. Proteins, essential biomolecules, form extended chains of amino acid residues linked by peptide bonds, adopting specific spatial conformations through interactions like hydrogen bonds, hydrophobic interactions, disulfide bonds, and ionic bonds. SDS, an anionic surfactant, denatures native proteins by disrupting non-covalent forces, including hydrogen bonding, hydrophobic interactions, and ionic interactions. Simultaneously, beta-mercaptoethanol acts as a reducing agent to break disulfide bonds. Additionally, SDS uniformly binds to proteins, rendering the intrinsic charges negligible compared to SDS-induced negative charges. This transformation turns folded proteins into linear molecules with a net negative charge, allowing separation based on molecular weights in a polyacrylamide gel. The gel forms through polymerization between vertically anchored glass plates in a cassette positioned between two electrodes. Positive electrode located at the bottom of the gel, whereas negative electrode is positioned at the top of the gel. After inserting the gel into a chamber, a running buffer is poured to facilitate current conduction. SDS is present in both the gel and the running buffer to ensure denatured proteins maintain their state throughout the run. Molecular weight size markers are commonly loaded onto the gel during separation, enabling the estimation of protein sizes from actual samples migrating in parallel in different tracks. After sample application, an electric field is applied across the gel for determining protein sizes in real samples migrating in parallel tracks. Each sample is placed in individual wells on the gel with bromophenol blue and glycerol. Bromophenol blue aids visualization, while glycerol enhances sample density and aids application. The electric field prompts the migration of negatively charged proteins through the gel, moving away from the negative electrode towards the positive electrode. Smaller proteins easily traverse the gel's mesh, while larger proteins are more likely to be retained, resulting in slower migration. Due to the small size of bromophenol blue molecules, they migrate faster than proteins. Optical control allows electrophoresis to be stopped before complete migration through the gel. After separating proteins, remove the gel cassette from the electrophoresis tank. Take off the glass plates, cut the top portion with the wells, and transfer the separated proteins from the gel to a membrane. In wet transfer, equilibrate the gel in transfer buffer, place it in a tray with blotting buffer on a rocking platform, and create a blotting sandwich by immersing a gel holder cassette in the transfer buffer. With the black side down and the white side up, and excess buffer drained, a fiber foam pad is soaked in the transfer buffer. Carefully placed on the black side of the gel holder cassette, a piece of filter paper soaked in the transfer buffer is positioned on top of the fiber pad. Once the gel has equilibrated in the transfer buffer, it is meticulously laid on the blotting paper. For the transfer, a pre-soaked nitrocellulose membrane or polyvinylidene difluoride membrane can be used. The membrane is placed squarely on the gel, followed by a second sheet of blotting paper wetted with the transfer buffer on top of the membrane. A second fiber pad, soaked in the transfer buffer, is then laid on top of the blotting paper. The gel holder cassette is closed and secured with a clamp. The primary method for protein transfer, known as electroblotting, uses an electrode assembly with positive and negative electrodes during the process. The gel holder cassette is inserted, ensuring proper positioning from negative to positive. The inner module is placed into the electrophoresis chamber, which is then filled with the blotting buffer. Following protein separation, cover the electrophoresis tank and connect the power supply to initiate the blotting. Electroblotting uses an electric current to pull negatively charged proteins from the gel towards the positively charged electrode, transferring them onto the membrane. The proteins move from within the gel onto the membrane while maintaining the organization they had within the gel when the run is complete. The electrophoresis chamber is disassembled and the inner module is removed. Then, the gel holder cassette is removed and opened. Next, starting with the fiber pad, each layer is removed until reaching the membrane. After the electrotransfer of the proteins on the membrane, the next step is detecting our protein of interest using antibodies. The membrane is first soaked in a blocking solution containing bovine serum albumin. To prevent interference between the membrane and the target protein-detecting antibody, it's essential to take this precautionary step. After applying the blocking solution, the membrane is placed on a rocking platform. BSA protein binds to areas without target proteins, ensuring that when the antibody is introduced, it exclusively binds to the specific target protein. Following the incubation period, the blocking solution is eliminated. Next, a tray is filled with a solution containing the primary antibody. The membrane is then gently agitated during incubation, allowing the primary antibody to bind to the specific target protein. After incubation, the solution with the primary antibody is removed, and a wash buffer is applied. The membrane is then placed on a rocking platform for additional incubation, undergoing multiple washes to eliminate unbound primary antibodies and reduce background signals. After washing, discard the buffer, add a secondary antibody solution to the container, and incubate the membrane with gentle agitation. The secondary antibody binds to a specific segment of the primary antibody. After incubation, remove the secondary antibody solution, pour wash buffer into the tank, and incubate the membrane on the rocking platform for multiple washes to eliminate unbound secondary antibodies. Thoroughly washing the membrane at this stage is crucial. After rinsing to remove any unbound secondary antibody, the membrane is soaked in a substrate solution. Chemiluminescent Western blotting is the dominant method for protein detection, with the secondary antibody typically linked to an enzyme like horseradish peroxidase. The preferred substrates use luminol and hydrogen peroxide, catalyzed by horseradish peroxidase, to produce the excited state product 3-aminophth phthalate. This product undergoes decay to a lower energy state, emitting light photons at 425 nanometers. The luminescence produced is directly proportional to the quantity of the horseradish peroxidase conjugated secondary antibody, thereby indirectly assessing the presence of the target protein. Following incubation, the membrane is covered with transparent plastic wrap to prevent drying. Densitometry is employed to analyze the membrane, with a CCD camera detecting the light signal and capturing a digital image of the Western blot. The obtained bands allow us to estimate the size of our target protein and ascertain its presence or absence in each sample. The introduction to the Western blotting method concludes here. Have you gained a clearer understanding of it?